Hello and welcome. I'm going to be talking about DNA replication in this video. DNA replication is where we take a double strand of DNA and convert it into two complementary strands of exactly the same DNA. Now to do this, we need to know a few facts about DNA. DNA is read from 5' to 3'. And since it's a double helix, we have two strands. And so for this, we have to unwind each strand using an enzyme known as helicase, which is shown here and here. An enzyme known as gyrase attaches here and here to lessen the torque on the rest of the DNA. And whenever we're replicating, this helicase unwinds the DNA and allows other nucleotide bases to join, forming strands such as this seen here. This joined 5' to 3' and is known as the leading strand, since it does not break, seen by these lagging strands that are short fragments known as Okazaki fragments. Now, whenever nucleotide bases join, they join in a specific pattern seen here. A always binds with T, and C always binds with G, and vice versa. Now I'm going to talk about DNA transcription. DNA transcription is making an mRNA transcript from a template strand of DNA. Now to do this, we have a structure called DNA polymerase that binds to a template antisense non-coding strand of DNA and start reading. This is in the initiation step. As it begins to read, elongation occurs and the nucleotide bases are complementary bound to an mRNA transcript. As before, A binds with T and G binds with C. However, in the mRNA, T is replaced with a U, known as uracil. And for this, we have a core promoter region, which is where the polymerase binds, and we have a terminator sequence, which signals the polymerase to get ready to stop transcription. We also have a transcription termination site, which is located directly after the terminator sequence. That is where transcription actually stops. And that is the third step of DNA transcription. Now, once we have our mRNA, it undergoes some changes known as editing, and introns and exons are within the mRNA transcript. Introns get removed because they are non-coding. However, exons stay and exit with the mRNA from the nucleus. Now I'm going to talk about translation, or also known as protein synthesis. We have our mRNA transcript here, and now we want to turn it into protein. So a ribosome attaches, and there are two units. There's a large ribosomal subunit, a small ribosomal subunit. It attaches, and tRNA, or transfer RNA, binds to the P site, which usually is an AUG first, which is a start codon as seen here, and this begins the translation. After something binds to the P, then the ribosome moves to the right, and another tRNA binds to the A site with an anticodon complementary for whatever comes next. In this case, it is CCC. And as this ribosome moves to the right, the tRNAs get moved into the P and then respective E site and then exit after that. But as it moves, it makes a polypeptide chain, which is a chain of amino acids, and as it grows, it forms a protein. Now there are 64 codons. 1 is start, AUG, and 3 are stop. They are seen here as UGA, UAG, and UAA. Now translation has the same 3 steps as transcription, which is initiation, elongation, and termination. And there are only 20 amino acids though, when we have 64 codons, so this means the code is degenerate, which means multiple codons can form different amino acids. And after we have our polypeptide chain forming proteins, and that is the building block of human life. Thank you.